Hi all, welcome back to lectures in microbiology. Today I'm going to discuss protein-based compartments. We're also going to discuss some endospore stuff. We're going to discuss a little bit about eukaryotic cells. So let's get started. First of all, remember back on previous lectures we talked about prokaryotic cells being unicellular by themselves, everything needed within that single cell to go about living. Okay, not like eukaryotic cells, although there are eukaryotes that are unicellular and function similar to prokaryotic unicellular organisms. But a lot of eukaryotic cells are multicellular, so you might have a cell that has the purpose of generating energy. You might have a cell that has the purpose of protecting the body. You might have a cell that has the purpose of lots of different things. Okay, but in prokaryotic cells or unicellular organisms, it has to be like within. And so we also talked about before that prokaryotes have no organelles, so they have no membrane-bound compartments where they can capture light and generate energy, or take glucose and break it down into energy, or package proteins. There are no membrane-bound compartments. However, what we don't often talk about is there are protein-based compartments. Okay? And so what a protein-based compartment is like these little rigid structures that occur in the cytoplasm or the cytosol. Cytoplasm, cytosol, kind of interchangeable, inside the fluid matrix inside this the cell membrane of the organism, and they have different functions. So let's say an organism is an aquatic organism. So here's a picture of the different structures. They kind of look like organelles. When you throw them under a microscope, they kind of look like these compartments, but there are no membranes around these. Okay, they're just protein capsules is what you can think of them as. And there's a better look at them. So they're kind of rigid protein capsules which can store or be utilized to do certain things. And some of them, like gas vesicles or these gas capsules, they allow for aquatic organisms to alter their buoyancy. Much like, for those of you who know things maybe about fish, and fish have, a lot of fish species have an air bladder, which is like a sac inside them, and they can take in different types of gases into that sac. Some of those gases are like gases that come off of cellular respiration. Okay, so sometimes it's carbon dioxide, sometimes it's nitrogen, other things. This is very similar, except for in a bacteria, and it's a protein coat, and they can deposit gases in that protein coat, which causes the bacteria to be more buoyant and float upwards in a body of water in a water column, and then they can expel those gases when they want to move down. Okay? And so again, kind of a useful structure that is developed in bacteria. There are other ones, lots of other ones that normally get the name or are abbreviated by BMCs, or bacterial microcompartments. Okay? Still similar structure, kind of this protein coated vesicle that in the case of BMCs, they can utilize these regions to carry out metabolic reactions, or at least to store the material needed to carry out the metabolic reactions. So sometimes the reaction itself, it can't occur in these little protein packages, but maybe the glucose needed for it, or the proteins that needed to be embedded into the membrane, like ATP synthase or something like that, or the proton pumps are, held inside these protein capsules until it's needed. So there's lots of ways at which this can occur. And then recently, we've found that even smaller than the microcompartments are nanocompartments, and they can do other things. They can hold other types of proteins that might be important for just binding up metals that are important for that organism's existence. So we know that there are iron binding proteins that occur inside nanocompartments. And so I want you to think of these similar to organelles, but again, they don't have membranes, so they're not membrane-bound vesicles. They are protein-based vesicles that have lots of different processes that they can help with storage and potential for, the ability for the organism to utilize them to move throughout a water column, all kinds of things. Okay. Now we're going to talk about kind of a unique structure in certain species of bacteria, and I think we'll probably find similar structures or similar like ways at which bacteria can survive as the endospore. The more bacteria we find, but the endospore is really unique. It's found in Bacillus, some Bacillus, and Clostridium. Okay, here's a picture of one, and really what this is like, you can think of it as this seed or this dormant structure. So what happened is, if you expose certain species to environments that they just can't survive in, like maybe it's extreme heat, or they're desiccated, so there's no water in the environment, extreme salt, whatever it might be, antibiotics potentially, like what that can do is trigger these different species of bacteria to go into the formation of a spore. So they'll form endospores or a endospore, in which they encapsulate their DNA. Okay, and so they would have that peptoglycan layer, that cell wall, and potentially, a capsule around that if it's, a capsulated bacteria. And they would just shrink their size. They also build, the peptoglycan layer is thicker and more resistant to different things. And what we figured out is they can sit in that kind of structure, and they, for the most part, they remove all kind of protein ports that would allow for access to the external environment. So they really have nothing. It's just basically like a cyst that forms, and they can go dormant for a long time. And we've documented some that we think are hundreds of years old, but others, we don't know how long they can last. But the desiccation one's pretty interesting. We've found bacteria endospores that were the result of like lakes drying up, and the lake hadn't been there for like 50, 60 years, and there's still endospores from the bacteria that lived in that lake, and they've just been sitting in a dormant state for a long period of time. That sporulation can be triggered by lots of different things. Carbon, nitrogen limitations could be triggered by, again, exposures to heat, exposures to all kinds of things. And the process, again, just to kind of go through it, so often, like, the DNA would be duplicated in case the condition gets better. And instead of the cell splitting and going through binary fusion, it kind of splits. A septum is formed between the two, and then an encapsulation happens, and so you get this encapsulated chromosome, and we call it a forespore when it forms, and it'll form inside the mother cell, right? And then if the conditions don't improve, the mother cell is lysed or degraded over time, and then you get this spore, this endospore that's formed. And that endospore, again, can be released into the environment, sit in the environment for a very long period of time. And we know that these endospores then can be triggered back into kind of forming a mother cell that has the ability to live in that environment by different triggers. So, and sometimes heat will trigger it, sometimes a different type of chemical, sometimes water can trigger it. It just depends on the environment. But it happens fairly fast. I mean, in labs, we can get, just by starving bacteria, we can get a sporulation or an endospore starting to form just by not feeding them. So really cool stuff. Potentially, obviously, hazardous to the medical field, because you might have thought you got rid of a species of bacteria, or you've been able to treat that species of bacteria, and we had an antibiotic that worked and it was great, and that bacteria could be dormant in potentially like fecal material or something like that for a very long time. Then if that fecal material is presented to a environment, or that spore, that endospore is presented to an environment in which it can germinate, then that bacteria can come back and wreak havoc again. So kind of cool stuff. Great evolutionary adaptation that allows for organisms to go into this dormant stage until conditions get better, and we see this in eukaryotes, but prokaryotes also can do it. Alright, so real quick, let's look at that eukaryotic cell structure. We kind of talked about this before. It's highly variable depending on what we're looking at. Like here you can see a plant versus an animal. If we threw a fungi on here, or a protozoan, they'd also look a little bit different. Of course, there are similarities. Similarity being they have a nucleus, they have membrane-bound organelles. Now, what membrane-bound organelles they have might differ between Protista and animal, and fungi and plantae, but nonetheless, they're typically much larger than prokaryotic cells. We often would say more complex, and when I say more complex, what I mean is there's more membrane organelles and compartments that are membrane bound for different features, for different structures. So complexity, not from the perspective of, oh look, there's more things named. That may be true also, but they carry out different processes in different regions of the cell with organelles, membrane-bound compartments. Okay. A couple key components. Protozoans, they're normally unicellular. From that perspective, I mean, there is debate on whether all protozoans are unicellular, but they're self-contained units, very similar to prokaryotic organisms, and they have a nucleus though, and no cell wall. Animals also lack the cell wall. Fungi and plants both have cell walls, but their cell walls are made of other things. Like fungi, it's chitin, and plants is cellulose, and that chitin cell wall kind of connects animals and fungi a little bit, because animals also produce chitin as exoskeleton material. And so that's for a different class, a different time, different discussion. Let's turn to how certain organisms might consume material or get material across that cell membrane. Okay? And this will really kind of tie in to a theory of how prokaryotes gave rise to eukaryotes. And so endosymbiosis, or the phagocytosis of another organism and then incorporating that organism, we'll talk about the next lecture. So we talked about this a little bit before. Endocytosis means that the membrane of the organism is flexible in the sense that it can engulf or take in material from the surrounding environment. Okay? If it's taking in liquid, we often call that penocytosis. If it takes in solids, we call it phagocytosis. If it's receptor mediated, meaning that it has these receptors on the outside of the of the cytoplasmic membrane, and those receptors are linked to some unit, like some kind of substrate that might be on the outside of an organism, or maybe it's just chemical molecules, like maybe just a bunch of glucose molecules or something like that. That's called receptor-mediated endocytosis, so they actually have receptors for that substrate. And when that occurs, it creates an endocytic vesicle. And all these cases are really endocytic vesicles. It just happens to be when phagocytosis occurs, we call it a phagosome. But there's really not much difference. And then once it's in, that membrane can break up, and that material can be lysed or utilized, however it might be utilized as an energy source, or whatever it might be. Okay. Exocytosis is basically the opposite effect. You have these membrane-bound vesicles, sometimes created by the Golgi apparatus, that will be sent to the outside of the vesicle, or outside of the membrane, and that those two membranes incorporate themselves or become incorporated, and the material is exocytosed out. This is a way that proteins can make it out of the cell that made them. This is a way waste products get ejected out of a lot of organisms. So it's not, exocytosis is not just for waste. It also can be for products that just need to be moved from one cell to another. Then maybe one cell exocytoses material out, and then the other one might have receptor-mediated endocytosis, where that material hits these receptors and comes into a different cell in a different region of an organism's body. Okay, so with that, we will come back and we'll talk about endosymbiotic theory, or how prokaryotes gave rise to eukaryotes.